So this is a continuation of the blood supply. Again, I'm using mostly my lab manual, Human Anatomy Clinical Approach. I'm mostly trying to make it so you can teach yourself anatomy, so you can learn what you need to know and find it out. We're dealing with the cardiovascular system. The lymphatics and blood play a major role in it, especially when we start talking about the respiratory, and the lymphatics actually when we talk about digestion. We can't take whole blood and spin it down, so we end up with 3 layers. We end up with the top layer, which is clear. There's about, 45 to 65% of the blood, which is called plasma. We have a little layer here called a buffy coat, and the rest of this is, with including the buffy coat, is the formed elements. There used to be a time where what they would do is they'd take blood, spin it down, and then they would get something which talked about what percentage of the blood from here to here, iron and red blood cells, and they call that a hematocrit. They still use hematocrit, but now they're actually looking at the iron and proteins in the blood, not the old method. Worked pretty well, but when we deal with plasma, the majority of it is going to be water, and then a lot of it's going to be proteins, and the electrolytes. These are your salts. So when you see a sport drink saying that they have electrolytes, pretty much they're telling you you're paying a lot of money for salt. So anything water soluble will be in the plasma. But what I'm going to talk about is the proteins. The main protein, about 60%, is a protein called albumin. After that, about 35% are what are called globulins. Then you have about 4%, which are fibrinogens. Then you have other circulatory proteins, which are sometimes are hormones. But what you notice is most of it is albumin. Now, albumin is a protein made in the liver. So even fibrinogens and some of the globulins are made in the liver. Almost most of the proteins are made in the liver. And this is going to be really important when we actually start talking about circulation. Electrolytes, again, you have your sodium, potassium, chlorides, magnesium, all the electrolytes. Anything with a charge is an electrolyte. When we're dealing with the formed elements and buffy coat, usually they're both the same thing now. We usually talk about the cells involved there. Now, the first one I'm going to talk about is your red blood cells, RBCs, which encompass 99.9% of all the cells. If you remember, our basic cells had a phospholipid bilayer. It had a nucleus. Now, the red blood cell gets rid of its nucleus. It has it when it's developing. When it's fully formed, it loses its nucleus. It has a cytoskeleton protein, a structural protein around it. Inside, you have 4 cytoskeleton proteins that are arranged in ring formations, hemoglobin rings. Inside, you put an iron. You do have an enzyme that's really important called carbonic anhydrase. Things that happens with red blood cells is it doesn't have a nucleus, it doesn't have the Golgi, it doesn't have anything that can repair itself, so it can only live a maximum of 120 days. Once 120 days, it dies, it gets recycled. The other cells you have in here can actually be broken down into granulocytes and agranulocytes. Granulocytes are cells with grains. Agranulocytes don't have grains. Remember, granule for grains, cyte for cell. A means without, so granulocytes and agranulocytes. You have a series of cells and cell fragments. One of the cell fragments is called platelets, which are what help you clot. You then have something called the monocyte. One site cell. This goes in a tissue. When it leaves the blood and goes into tissue, it's called a macrophage. Macro means big, phage means to eat. So macrophage is to eat big. These cells are going to eat whatever is not supposed to be in the body. They're non-specific. They're part of something called the non-specific immune system. Lymphocytes. They're specific immune cells. They actually look for specific things in the immune system. And they can be your T cells or your B cells. Now, one of them is called a neutrophil. Phil means love. Neutral means neutral. So it loves something that's neutral. It has 2 types. It has granules that are basic and granules that are acidic. So it has a mix of these different types of granules. What it actually does is it tends to eat bacteria. It's not specific. It just sees that something's not supposed to be there. It eats it. Usually these have multiple lobe nuclei. So you actually will see nuclei that look kind of like a jumble with a bunch of holes. These neutrophils are part of the non-specific immune system. The other cells that we have are basophils. Baso is for basic. Phil means love. So these are cells that love basic dye. And the dye that's used to stain these tends to be blue. So basophils have really dark blue granules all over them. And what they do is they has histamine. And histamine, when it gets released, causes swelling and calls things to the surface. These are involved in a lot of the immune response, the systemic one. So these will actually call cells to an area and cause the vessels where these cells are to become leaky so they can get in and out easier. The last one is called the eosinophil. Phil means love. Eosin is a acidic dye. So eosinophils have these red dye, and they're really phagocytic cells. Phago means to eat. Phage, cytic means cells, so these are cells that eat. And they are actually also responsible for fighting off parasites and are involved in allergies. And so those are the main cells that you're going to actually find. I do want to tell you guys about what are known as the cardinal signs of inflammation. And you'll get this in physiology. Cardinal signs of inflammation are what you get when you're sick. First, you have heat, which we call calor. Then you have swelling, which is tumor. You have pain or itchiness, which is dolor. And you have a redness, which is rubor. Now, if you speak Spanish, you've come across these words before. Calor, heat, tumor, swelling. Dolor is pain. Rubor is a redness. The reason you're getting redness is, like I said, the basophils are degranulating and causing more blood to come to the area. You also have the swelling because it causes the capillaries to become leaky. With these cells degranulating and eating stuff, you actually do have some heat. Also, there's some response that your body does to the hypothalamus to increase heat. A less favorable environment for anything that likes to go there. Pain tells cells that there's damage, and so it tells it to go to that area. And then you can actually have system dysfunction, which means that you've actually gone to a point where things are breaking down. So those are the cardinal signs of inflammation. I want to tell you about how they develop. Everything's going to start in the bone marrow, and we're going to start with the cell type called a pluripotent hematopoietic stem cell. Now, don't be afraid of the name. Pluripotent hematopoietic stem cell is a sentence. Pluri means many, strong, hemato for blood, poietic making stem cells. And it starts and it divides into two types of cell. One of them is going to be called a myeloid stem cell, and the other one is going to be a lymphoid stem cell. And I'm going to talk about the myeloid first because it's one that makes most cells. This will divide into three types of progenitor cells. One of the progenitor cells will end up making a cell, and it'll go from a prourethroblast to a erythroblast. Eventually, it will get rid of its nuclei, and what you end up with is a erythrocyte, which is called a red blood cell. You can make a cell called a megakaryocyte. Now, you've heard the word eukaryote. Karyo is the same thing, so it's actually a cell with a nucleus. Megalokaryocyte will have a big nucleus, and then it'll break apart into a bunch of fragments, making platelets. The next one is going to form what's called a blast cell, and the blast cell here is going to split into two different types of cell. You're going to make one which is going to be a monoblast, which will end up forming a monocyte. By the way, you can see mono and blast means maker, so monoblast. The other one will end up making the myeloblast, which will make three types of cells. These three types of cells are going to end up eventually becoming the basophils, which I'll put B in the heart because it loves base, eosinophils, and neutrophils. So this is actually what the myeloid stem cell makes. The lymphoid stem cell is going to make lymphoblast, which are eventually going to form lymphocytes, which can end up being either a B cell or a T cell. And that's how they form. Now, all these cells are going to be made in the bone marrow. B cells start in the bone marrow, and they're educated there. Pretty much they go through a series where they can make any immunoglobulins to anything, which gets blood from the superior, inferior vena cava, and also the coronary sinus. It goes to the right ventricle, where it goes through the pulmonary trunk, to the pulmonary arteries, to the lungs, where it picks up oxygen, and it comes back to the left atrium, to the left ventricle, and the left ventricle goes through to the aorta, and then the aorta, it ends up branching into large arteries. So you have your aorta, to large arteries, to medium arteries, to smaller arteries, to arterioles, and from arterioles you'll actually have a specialized one called a medarterial, and then it's going to branch into a lot of capillaries. Now every time this divides, you have more tubes and more space. The pressure goes down and down until you get to the medarterioles and capillaries. The capillaries will end up having almost no pressure. As you go down, the arteries become smaller, but also the tunica muscularis of the artery becomes smaller, to the point that when you're in a capillary, you have a single layer of flat cells in a tube. So if you were to turn this around, you'd see a tube here and all the flat cells coming across this way. Now, we talked about this type of tissue before. Flat cells in one layer is called simple squamous endothelium. This time it's inside. Now, what ends up happening is all the cells can pass here one at a time, and you have millions of these capillaries inside tissues, but they have to go one at a time. Now, there are problems that can happen. For instance, there's a genetic condition called sickle cell disease, where there's a change in one of the amino acids, and if it's low oxygen concentrations, it changes shape to become a sickle, and it can get stuck on the capillaries, causing a sickle cell crisis. What ends up happening is you're in a tissue with one capillary here. Things can move out of here and in here, and it's based on concentration differences. So you're dealing with osmosis and in here, and it's based on concentration differences. So you're dealing with osmosis and diffusion. You're also talking about pressures caused by fluid moving across, hydrostatic pressure, and you're also, which is going to push stuff out, and you also have colloid pressure from the proteins pulling stuff in. So proteins make sure the fluid between these two areas are about the same. Now the area in here is called your interstitial space. So what happens is, as we're dealing with diffusion, water can move across based on concentration differences. As you have proteins, it tends to, usually under normal conditions, be relatively same amount of water in, same amount of water out. You then also have oxygen. Now, if this is in a capillary in its muscles, you're going to have more oxygen in here than out here, so it's going to be able to diffuse out. Carbon dioxide is going to be able to come in. Metabolic waste, like nitrogen waste, ketone bodies, is going to be able to go from the tissue into the blood vessel. And then it comes together in the forms of venules, then small veins, bigger veins, until you actually get to the vena cava or the coronary sinus, and then you get back to the heart. And so that's actually how this works. Now, the reason that all the blood is important is this. First, you have a capillary bed. If you have an infection in this area here, cells get called to the area, and the degranulation causes the capillaries to become leaky. So macrophages can end up sneaking out. It also causes a swelling in this area. We're going to have swelling in this because of immune system. The other thing is, you're not a stationary being. You're constantly moving. And so sometimes when you stand for a long period of time, the fluid in the capillary builds up and it moves out. Now, the reason that happens usually when you stand is in your veins, you have one-way valves. And every time you contract muscles, you tend to move the blood. There's actually a muscle that's very important for the cardiovascular system to help the blood in the lower area continue to move up. And that's your gastrocnemius and soleus, what most people call your calf muscles. As those contract, it pushes blood up. That's why you have some people who, when they're at work and they have to stand all day, their legs swell, but they can go on 5-hour hikes and their legs don't swell from that. What that means is the proteins are really important to make sure fluid is also pulled in. So if you have liver problems, as you're not making the plasma, as many of the plasma proteins as you normally would, you would actually end up having leaking. And that's one of the things that happens with liver problems. One of the first signs is edema. You have fluid in the tissues. The other thing I wanted to tell you is most people think that oxygen comes in and carbon dioxide binds to red blood cells. The truth is, oxygen comes into the red blood cells, binds to hemoglobin, and you can bind 4 for each iron because you have 4 hemoglobins. But the carbon dioxide is CO2, will actually bind temporarily. And you have an enzyme in red blood cells called carbonic anhydrase, which will take water and it's going to bind them together. And what it will form is H2CO3. This is called carbonic acid. And carbonic acid, pretty much, as it's an acid, hydrogens will come off, and you'll end up having free hydrogens and bicarbonate. If this is in the red blood cell, this hydrogen will bind to the hemoglobin, and this bicarbonate will go into the blood, which is good because the pH of blood is going to be roughly 7.4. There's a range 7.35 to 7.45. And that's called physiological pH, which is a little more basic. Part of it has to do with this. The other thing that makes it more basic is the proteins that you have. One of the neat things is this acts as a buffer because you also have carbonic acid in your blood. So you have carbonic acid and you have bicarbonate. What happens is this extra hydrogen bounces back and forth. And so what happens is if your pH becomes more basic, the acid will end up giving you the hydrogen, so you make it more acidic. If it's too acidic, the bicarbonate will neutralize that. This is a buffer system, and it's really important to understand, especially when you get into physiology and respiration. We now actually have an understanding of how the blood is going to work. I still haven't given you respiration part, just the basics here. When I talk about respiration is how this works out. Now, I haven't also told you where the carbon dioxide and water comes from, which will come in when we do digestion. So this formula you're going to see over and over again. At the lymphatic ducts, you also have something called lymph nodes, and these lymph nodes are attached to one of 2 draining systems. One of them is called the thoracic duct, which runs parallel to the vertebrae and then drains into the subclavian vein, which goes into the brachiocephalic. The other thing, though, is you have only the lower portion of your body and the left hand use the thoracic duct. The right side uses the right lymphatic duct, which what it does is it pretty much drains the fluid from the right arm and chest into the subclavian. Now we actually understand how all this comes together. There are 2 things I haven't told you about, and that is development of the cells occurs in the bone marrow and then it leaves. T cells go to the thymus. The other ones will go into circulation and lymphoid tissue. Now, if you are to look at bone marrow, what you notice is a bunch of cells with nuclei. In blood, you don't have as much nuclei, and the reason is in the development in the bone marrow, the nuclei of the red blood cells are shed. You don't have nuclei in your red blood cells. See as a main difference between the two tissues. Most tissue of the lymphatic system is going to be a tissue type called reticular, which is pretty much a bag tissue. Mucus-associated lymphoid tissue, a BALT by the way, which is bronchus-associated lymphoid tissue. What you have is you have a center in the middle, and you have aggregated lymphoid tissue around it, and it's clear in the center. You also have tonsils, and tonsils, you usually have lobes, you have some connective tissue running there, then you see areas where they seem kind of clear. Those are the germinal centers. So again, you see similar things in both types of tissue. As we continue, what we end up seeing is we can end up looking at lymph node. And in lymph nodes, what you have is you have a pretty much connective tissue around it, forming a capsule. You have an area for an artery to come in, I'll make it red. You have an area for a vein to come in, I'll make it blue. And you have an area for the lymphatic drainage to leave. So from this lymph node, you have an efferent or capillary. Efferent means exiting. You also have afferent capillaries, which are bringing fluid in. So all the fluid that was leaked out of the regular capillaries are picked up by lymphatic capillaries and brought into the lymph node. In the lymph node, you have a lot of different areas. These will actually go deeper in. You have an area here, which is called the paracortex, which is full of T cells. You then have areas here, which are kind of clearer, which are called your germinal centers. In these centers, we have B cells. Now, the thymus pretty much has the same look. What I'm going to do is draw you spleen quickly. And the spleen, what you'll actually see is you'll see a capsule outside. You'll see these real pale circles, and you'll see these areas here. These circles are called white pulp, and the areas between them is red pulp. But what you're seeing is in every type of lymphoid tissue, you have this arrangement. You also will have capillaries going in here. And one of the neat things about the spleen is attached to the hepatoportal vein. The reason I mentioned the spleen and the hepatoportal vein system is this. In the spleen, you have a lot of lymphocytes, a lot of cells that can eat cells that aren't supposed to be there. And one of the things that happens is when red blood cells get old, the cytoskeleton starts popping out of the cell membrane. And these macrophages and tend to see that the cytoskeleton starts popping out of the phospholipid bilayer. And if it happens in the spleen, it can go from you break down the hemoglobin ring, making something called unconjugated bilirubin, which means it doesn't have any sugars, so it's fat soluble. And the unconjugated bilirubin will go from the spleen directly into the liver through the splenic vein, hepatoportal vein, liver. And in the liver, it'll be conjugated, which means I'll put sugars on it, and therefore it'll become water soluble. And you can get rid of it easier, whether it's through the GI tract or renal system. And that we'll talk about when we get into digestion. Thank you. I hope you enjoy this. If you're feeling lost, you may want to try to get the book. If you're not in a hurry to get it, I would wait until September 2017 because I do have a new edition coming on. Usually I use this for my class, and so they need to have it pretty regularly. So I hope you enjoy this, and I hope this makes sense to you. If there are any questions, please write them down. I am also going to do a lot more videos in martial art, which will come to talk about anatomy. So a lot of times I'm going to try to refer people back to these videos. Thank you for your time. Goodbye.